first component of IPM is resistance. Means we develop the resistance line, we develop the resistance genotype, or we can induce the resistance in the plant for controlling the insect pest. That's why I choose this topic, and this is the drawback. That's why we use that chemical insecticide is the major problem, and we are using mostly in all over India, that mostly 90% using our chemical control, no using IPM method or resistance or induced resistance. And that's why many problem is there, like economically also many problem due to this chemical insecticide, environmental effect also there. But if you using our resistance work or induced resistance, that may be easy or very good for future for organic or your natural farming also very good. So what is the mechanism of resistance? This one slide, that why requiring this resistance and what are doing this mechanism. So in this three part is there. One is antigenosis, another is antibiosis, and last is tolerance. Three part is there for mechanism. Any, if you are any line you produce, any genotype, any plant, if you are any is there, that some mechanism is there. So in which that first is antibiosis, means some chemical like tannin, phenol, phenolloid, different type of chemical is found in plants that is resistant to against the biotic factors. Then second is antibiosis. This is some morphometric character, morphological character. This is also antigenosis. So many type of character like hairiness and leaf hardiness. So this is also there. Then last is tolerance. Sometimes insect is attacking on plant, but no any impact on this yield of the plant or of the crop. So this is also important. So these are the three major mechanisms. That's why we require this resistance. So my study, I work on rice against the aphid. So in this we have using for induced resistance. Induced means we apply some chemical like silica, metarhizium, vivaria, trichoderma, PGPR, different type of PGPR also there, that immunity, increase the immunity of the plant. So this is a most of the word, and I have seen in Thailand that one silica, one separate journal is there that publishing around a 10 to 12 impact factor is there. So separately, so most of the that work on silica, silica inducing the resistance in plant, and after that they controlling the biotic as well as abiotic, both are controlling in the plant. So that's why we use the silica as a induced chemical for resistance against aphid. And these are the already some reported scientific reports and other, that China and other already work done on this silica work. So what we start that for, we start the one experiment that we using the 2% calcium silicate, 4%, and one is control. And after that we started two experiment. One for that enzymatic activity, what is the effect of enzymatic activity after that applying your aphid infestation and as well as your different treatment. And another is estimation of silica in plant. This is also important, because after that 50-60 days after we identify that how many percent of silica that deposit in the plant, different treatment work. So this is also very good result is coming. Another we also taking this aphid data of this rice. So after that we, after applying this in initial stage your 2% or 4% calcium silicate, that is silica, so we release the aphid in the after 20 days seedling, and we see the enzymatic activity first. What the effect of enzyme when applying this silica as well as applying of the biotic means your aphid. So this after we taking the data before the applying, then after 24 hours, 48 hour or 72 hour, and then we found the difference in enzymatic activity of the different POP, SOD, that catalase, different type of enzyme is there that affect due to this biotic stresses. So this is, we have taken all the enzymatic activity in the lab after that treatment of all this 2%, 4%, and infested, non-infested, that all data we have taken in the lab. Another experiment is on silica. Silicon, we apply same 2% silica and 4% silica, and after that we see the difference of aphid infestation. If I see next slide, that if you see that in control that highest infestation is occurring, we have released same population of aphid, but in control that highest incidence is found. But in your that T2 or that 2% or 4% T1 or T2 treatment, that having very less incidence of your that aphid infestation. So this is also important. So after that, we take this data, and you see directly that control having highest incidence of this aphid, then followed by this 2% or 4%. But we found that 2% and 4% are non-significant, not significant, no difference. So after finally we recommend this 2% is better for inducing the resistance in the plant of rice against aphid or also another biotic factors. Then after around 60 days old plant, we take the leaves of different treatments of silica plant. Then we go in electron microscope. This is very good facility in this Prince of Songla University. In one that faculty, separate faculty is there. Central laboratory is there, that having 4 electron microscope. So we take the that data of silica, how many percent of silica is deposited in leaves, because that is directly correlated to resistance. If you are more silica is there, then your cutica or that plant cell all are hardy type, and your tannin, phenol, and flavonoid also increase. So that why resistance is developed. So this is the induced resistance. So we found that after that we taking data, you see the condition of this. In control, that very less amount of your silica. But if you see 2% and 4%, then increasing around, I see in next this slide, in control that having only 7 to 7.5% that silica content, but in T1 that having 13.5% silica content, and another that in the T2 control 4% that having around 15%. So in 13.5 or 15 that is no non-significant is found, but both are increasing from the control. So clear cut result is coming from directly. No need to any chemical and other. Directly we use the leaves and entering this microscope, and directly that result is coming. That not only this silica, also oxygen, carbon, how many percent. This is also impact. Increasing your silica, then your carbon and oxygen also decreasing. And then also another impact also we have seen that maybe potassium, that also increasing with the silica content. So this is also positive result we found. So outcome of this last 3-month training, we have already make one paper that silica acquisition and accumulation in rice cultivar confirms resistance against aphid. So that may be coming in a good journal in future. So already that running. So we can, for future plan, we can use that in, because in India the rice is the major crop, so we can use for biotic stresses. Many type of biotic stresses is there. Already China in working on leaf folder already done, and then aphid we have started new. So this is also important. Another, we can use the safety, because if you are applying chemical, this is not good for like organic and natural farming also there. So, and we can also use PGPR, different type of PGPR as well as metaerism. We seen one that paper that also metaerism or your trichoderma, that is also increase or improve the immunity of the plant. So this is also good. So another, when I joined in 15 April of the Prince of Songla University, so after 10 days, one training is there. For 5 days, that is collaboration with the European Union. That project is funded by that European Union. So it's also very good and totally on entrepreneurship because I seen in last 3 months in Thailand, mostly all that university is working on entrepreneurship because after BSc, MSc, where is go student? So all are working on entrepreneurship, and mostly I see 90% student BSc of agriculture that not going to MSc or PhD, only going your personal entrepreneur. So that's we have also 5 days that all visit and other there. I have seen a different type of entrepreneurship and also discussing different type of person is coming from all Thailand person and present this training. So this is also very good opportunity give me. So in this all that and in this online also that European Union and that another country also join this. So that completely work on this entrepreneurship. So this is also important. And after that we going to in villages. So we found that one place that homestay. So this is also important. All raw material and all food is available locally. That is available this hotel in the farmer field and all that person is coming in this all food and all that locally or organically available and given to the persons coming this hotel. So this is also important. So this type of that committee is there the all and then promoting in all the that Thailand. Yes sir. Mute sir. Who is this? So another we have found that that's a coffee farmer. So this is what he do. Actually in India most of the farmer raw material selling to the market, but in Thailand this is not there. All farmers going up to the post harvest or ending marketing also done by the farmer. This is very good that's why maybe economic condition also very good of that Thailand farmers. So you see that farmer given the training of coffee that selling also product and in field that coffee also available. So all material in one places is there. Another I have seen this mulberry plant. That one farmer is there that having mulberry, and that you see that having this product, and then selling in market also, and then display also. You see this. So this type of work is that is working in Thailand. Mostly that final your product is given, and no any marketing person and other. That's why that maybe economic condition also good of the farmers. So this is the farm and that all that
किसान का फार्म है.
So another we have seen the entrepreneurship through honey. Honey bee also important and this is stingless bee. Most of the I see farmer 4 5 farmer. So most of the working on stingless because in India we have only some farmer also in Manipur also there. I seen some one cruel that having one farmer that producing stingless bee and it's producing only half around 200 to 300 ml per boxes, but cost is very high. In Thailand, that cost is 5,000 to 6,000 per liter per that is very high costly, and that is medicinal value is very high. That's why that rate is very high. In India, around 2,000 to 2,500 in India also there, but Thailand is more than our India. So this is also important, and then easily reared. No any sting is there, and but is producing only small. And this is that BSc student they starting this business.
ประมาณ3 รังนะ
So before that father is working on this
เนี่ยไซ
honey
ส์
bee.
ไซส์คือไซส์
So
มาตรฐาน
after
เมื่อ
that
กี้
they start the online business, online marketing of the honey and byproduct also different type of. So you see this. This is the rearing method and making this type of boxes. This is stingless, only producing around 200 to 300 per boxes. Another I see one farmer that having around 100 that boxes. You see this all boxes of stingless bee. Because that rate of this honey is very high, 5,000 to 6,000. That may be mostly working on this. This is stringless bee, but the species may be different from India. That is there, but it's a very good farmer. That you see all complete. That stand also there. All there. In Nagaland, our India also that Nagaland, that area that some farmers working on this stringless bee. So another is edible insect also important, and you see the market that all type of that like cricket or beetle, all is there. So this is also very good that entrepreneurship in Thailand. In southern Thailand, so that is major because in southern Thailand mostly eating that insect as well as seafood. You see, many market and many soap is there on this edible insect. Another that we have seen protected cultivation. One farmer that also working on this protected cultivation, and you see the this that different type of cooker beets is there. So that also growing. Another important I seen that maybe 20 that my mentor is told that in 2080 maybe that two center is open in Thailand. One is southern India or another is northern India. That name is PSC means Science Park. One southern science park, another is northern science park. That main role is entrepreneur means that university develop our technology that all selling, marketing that do by this center. But this is a very big center. You see all that all product of that PSU because PSU is having around 10 to 15 different center also there different districts, different that state also. So that's why that all that colleges and all center send here and that marketing private companies coming and that giving the MOU and all work do this. And simple technology. I seen that methyl that is available in market but that market is available, but farmer don't know okay, what is the use of this, that what will we do. So for that, they are making the pheromone trap and then give it the farmers. And then making the technology simple. That rubber is using for applying. So market, they purchase the pheromone and they give. Another I have that conducted one training, my mentor. So that also like trichoderma, we are working on last 50 years. But that going up to the farmers level. They start that is mother means start from that na that training means that not given by voice or other. That given the raw material and start the complete procedure. This is maybe good opportunity because without sin that may be not possible. So on the basis of that entrepreneurship, I thought some work is due. So I written one that review paper. That sustainability one journal is there that may be accepting this journal, and also I written one book also on entrepreneurship. So another teaching method. So I already told that all you see that scientist is there. Also, all mushroom that is maybe medicinal mushroom is there. All display on the table, and all student, all that farmers is coming and seeing which is better, which is so. This is very good method for teaching. You see, this is the display also, and there are also all that packaging and all the technology, all is available.
เก็บเห็ดมาสดๆเลยเนี่ยทำสต๊อกก่อนเลยค่ะเพราะว่าหลังจาก
We working only on this medicinal mushroom. What different around 100 to many type of that
บันทึกลักษณะไม่ใช่ว่าไปเก็บ
mushroom
เห็ด
is
มา
there,
วันนี้บัน
medicinal
ทึกลัก
mushroom.
ษณะพรุ่งนี้นะคะเพราะลักษณะมันต่างกันแล้วนะพรุ่งนี้อาจจะเป็นลักษณะที่เป็น
So another I also delivered some lecture to PG and PhD student, UG also there on honeybee as well as that botanical pesticide on entrepreneurship. Another I seen that farmer market. This is important. That maybe in India we started that may be very important. Actually, alternate day of that PSC Prince of Songla University that evening 2 to 7 o'clock. That 3 to 4 hour that open. all farmers, local farmers coming and our raw that material they taken here and then fresh fruit given to fruit, vegetable and all products. And also that university also display our product. So understanding also between farmer and that university also there, and that may be good for that displaying or technology farmer as well as our that university also. So this is maybe good for future, and this also appreciated by that state that union, European Union. This also very impressed on this farmer market. Another this campus landscaping that is very good. You see these are the not private buses. This is university bus, and all this is president. Actually, in Thailand, that VC post is equivalent to president. That name is president. So he is president, and that one that program is conducting of around 10 to 12 all universities. So that is you see in inside also. This is the big auditorium that is there. So all facilities there are very clean, and all that facilities may be good. So we also applied for that MOU with this university. So my suggestion for university is develop the farmer market at university level or at college level. It may be good initiative for India as well as our university also. Then promotion of Indian farmers for development of own processing product, because this is important. Most of the our farmers given the raw product, means given the rice but not given the byproduct or that post harvest. This is maybe good for extra income as well as reduce the middleman, because middleman also most of the that middleman is also taking, so that maybe also increase the income of the farmer. Then we can also another develop the central laboratory. This is important. In PSU Prince of Songla University, that separate faculty, means here we call that college. In that Thailand, that name is faculty. Same. That boss is dean. So this is also separate faculty is there for instrumentation. All instrument is there. We given the 1000 baht and given the all this picture of electron microscope. So maintenance also good, and then technician also there. But here we take one scientist, we have that instrument, but another also there, but no any maintenance. So that may be very good work. Then another entrepreneurship development for agriculture graduate student may be good for future. Because most of the that after BSc, no one is going for MSc and PhD, only limited. If professor is requiring any student, that some student is coming. But no one is that MSc and PhD, only BSc, after that start your own business. Thank you. Thagatsari and Swadikar. Any suggestion, discussion?